Okay, well, thanks everyone, and hope you enjoyed the coffee break. So let's move on next to the third lecture that we have today in the series. And I'd like to then be introducing Dr. Stephen Ogle, who's a professor in the Department of Ecosystem Science and Sustainability at Colorado State University. He's going to be talking about measurement and modeling of soil carbon and soil greenhouse gases. Steve's been at Colorado State for a number of years, working on these issues. One of the main parts of his program now is compiling the estimates for carbon and greenhouse gas emissions from soils for the U.S. National Greenhouse Gas Inventory that gets submitted by EPA to the UN Framework on Convention on Climate Change every year, basically tracking then what's happening in the U.S. in general in terms of greenhouse gas emissions and emission reductions. In addition to that, Steve has worked on and has a number of other different projects involving a combination of field work and modeling and use of remote sensing and things like this. So Steve, please come on up and we're eager to hear what you have to tell us about a central issue of the whole workshop here. Thanks.
Well, thank you, Keith. Yeah, it's really nice to be here. I'm happy that organizers invited me. This is a topic that's near and dear to my work. I've been at CSU actually over 20 years now, almost 23 years. I am an ecosystem scientist by training, and as Keith mentioned, I work a lot on doing inventories, particularly at the national scale, so how we quantify. I think Dave mentioned this morning in his talk, we can't manage it if we can't measure it, and that's been directly part of my work over the years, is figuring out how we can measure it or modeling it in most cases. So I'll talk more about that in a moment. So I want to give you a little context to start out why this is important. Some of that's already been given to you, but I want to go into that a little bit more now. And then I want to talk about measurement-based approaches and model-based approaches to quantify soil carbon stock changes and greenhouse gas emissions, methane and oil, but I'm mostly going to focus on soil carbon today. Don't really have time to talk about everything. So why do we want to enhance agricultural soil carbon stocks? It was brought up earlier today about trees and forests. We can see those, right? We know the carbon can stay in those trees for decades to centuries, right? Very easy to go out there and measure those at diameter at breast height, how tall those trees are. We can use allometric equations to get that. We don't have that in our agricultural systems. We don't have trees. We don't have that woody biomass that's going to stay there. Those annual crops, they're going to die every year. If they're perennial grasses, there will be some storage below ground, but there's not as much storage as you're going to see over time when you have trees. But soil carbon can lead to long-term storage. This is a paper from Shi et al. In which they were showing that the age of soil organic carbon can be quite long, particularly when we get in the subsoil. So if we can increase the carbon in our soils through management activity, that can lead to long-term storage, which is what we're looking for. Not storage in, say, a corn plant that's going to die that year, and most of that CO2 is going to return to the atmosphere in a matter of a year. If this can get into the soil, then it can be stored there for a long period of time. Other greenhouse gas emissions. In the U.S., this is from the U.S.'s National Greenhouse Gas Inventory, almost 75% of N2O emissions in the U.S. are from agricultural soil management. That's the way we manage nitrogen for the most part. So a tremendous amount of emissions. If we want to deal with soil N2O here in the U.S. and many other countries, we're going to have to look at agriculture to reduce those emissions. Methane, similar with rice. Not so important here in the U.S. We don't grow that much rice, but in other countries, particularly in Asia, there can be quite a bit of emissions from rice. If we want to deal with those emissions, we're going to have to do it in agriculture. So a key challenge here is quantifying that benefit. How much sequestration are we getting? How much N2O emissions and reductions are we getting, methane, et cetera? And I think of this from the standpoint of doing greenhouse gas inventories from the IPCC guidelines. And when we're quantifying that benefit, we also need to quantify the uncertainty in that. And I don't mean doing a sensitivity analysis. Many of us, I'm sure in this room, are modelers. You can take your model and vary the parameters, maybe plus or minus 5% or some other way, and you can see how those parameters influence the result. And that's valuable to know. But that's not an uncertainty analysis. When we do an uncertainty analysis, we need to be drawing an inference that we can say at the end of the day with a confidence interval, how likely the true value, what's the likelihood of the true value. For example, if it's a 95% confidence interval, it'll fall between this range, and you can see some of those here. This is from the EPA's report from 95 to 2015. You can see those ranges are pretty large. This is for soil carbon sequestration in croplands over these years. So one of my main hopes this week is that we're able to find strategies that we can reduce that uncertainty. And why does that matter? If we think broader about this for a moment, the U.S. government's plan to reduce emissions by 50% by 2030. It's not so far away. And there's a lot of different strategies they're taking to do that. And sequestering carbon in agricultural soils and reducing into a methane from agriculture are some of the ways that they're approaching this. If we look at this uncertainty here, this is from one of those years in the previous graph, it's 2011. That uncertainty ranges from about 5 to 25 megatons of carbon. Of course, they're hoping to be sequestering more carbon as they move forward with these programs. That's quite a large range, and it could be that the level is closer to the 5 than it is maybe to the 25. Or the estimate that they would use would probably be about 15 in this case. So if they're adding up their totals, using 15 in this case, to see if they're achieving their goal of reducing emissions by 50%, there's some risk here. That 15 may not be the amount of carbon sequestration they're getting. And as we think about the Paris Agreement and trying to meet our commitment and keep warming below 2 degrees C, this is very important. We need to reduce these uncertainties as much as we can. Okay. So, how do we estimate soil carbon stock changes and GHG emissions? As I mentioned, we can't just walk out there and measure. There's no, I think Keith recently said there's no thermometer or something we can stick in the soil and here's the amount of carbon. We have to use techniques where we can quantify this over time. It's not easy. As Dave mentioned, there's not many soils measurements around the world to begin with, or we can model it. And I'm going to talk about both. I'm going to start with measurement-based approaches. Here, there's really 4 steps to this. I'm going to go very basic here, as I was instructed for this lecture, is stay very basic. This should be the 101 level. I don't know if I'm quite at the 101 level, but I'm going to try. So with measurement-based, the first thing we've got to think about is a sampling design. Where are we going to take these samples? We've got to think about the methods we're going to use when we get to the field. We need to think about what we're going to do when we get back to the laboratory, and then finally be able to calculate those soil carbon stock changes or GHG emissions. So let's start with sampling design. Basic sampling design here. If we want to draw an inference on agricultural lands, let's say in the US, then our population is all the crop fields and grasslands in the US. That's our population. And that needs to be part of our sampling frame. In fact, the way I drew this here, the sampling frame wouldn't include that entire population. You see there's some edges there that aren't covered by that sampling frame. That sampling frame then is where we're going to draw our samples. Every field should be available to be selected in that sample when we do this. And then we'll draw that sample. And we can then sample in different ways. Just to be familiar to most of you, we can do simple random sampling. We might do simple random sampling of that population if we don't know much about it. That would probably be as good as any other method. But oftentimes, we might want to space those out because we know there's variation across geography. And a systematic random sampling would be a way to do that. We could space those out, randomly locating that first sample, and then basically laying those out in geography. In fact, the Lucas system in Europe is set out that way, a systematic sample. We could also use a stratified random sampling. Oftentimes we're sampling soils because we know something about the soils already and how they vary by climate, by soil types, topography, etc. We're probably going to use a stratified random design in a lot of these cases. And then maybe within those strata, we're going to do a simple random sampling or systematic. Scale is very important as we're thinking about this. If we're sampling a field like this canola field in Georgia, we're obviously going to have a lot of samples in that field, maybe 50, 60 samples in that field to really quantify what's going on there in terms of soil carbon. If we're sampling the entire country, this field may not even be in the sample. If it is, we're probably going to take one sample there. We don't care so much what's happening in this individual field. We want to know what's occurring across the entire country. So we're going to have samples laid out, dispersed widely across the different types of geographies, climates, soil types, topographies, etc., that occur in the country. So that's a little bit about the sampling design. Let's think about what we're going to do in the field. One of the issues when we go sample soils, I know many of you are going to be familiar with, if we go today and if we're in a field right now and I sample right here, I can come back a week later and move over a few feet and take another sample, and there's going to be a difference. Now that doesn't have anything to do with a difference in soil carbon stock change over time. It didn't change that much over that week, but it's just because of heterogeneity in soils. And we have to control for that heterogeneity when we're sampling in the field. We don't just want to go out and take one sample in the field. That's not going to be enough to get a good estimate for that field. And one of the reasons, is because there's this phenomenon known as regression towards the mean. And so if we go sample, let's say in this graph here, we sample a higher baseline stock, say this soil has a range, and we sample on the higher end of it, there's a good probability next time we come out and sample, we're going to be sampling down on the median part of that range or the low part, and that's going to make it look like there's a change in soil carbon when we're just sampling the variability in that field. Each time we go sample in that field, we need to get a good estimate of the mean. We need to manage that variability. And there's different ways of doing that. This is one sampling plot that we've worked with USDA, and it has a triangular design where we take 9 samples each time we go out. We start in the corner of these 3 smaller triangles you see here, and basically we're moving clockwise around those triangles over the years. And it might be, as Dave mentioned, we're going to sample every 10 years or maybe every 5 years, depending on how much change we're expecting. This particular design you can sample for about a century. So a long time frame in this. And each time we're getting these 9 samples to get a good estimate of the mean at this location and not just sampling some part of that variation when we're out in the field. There are also other considerations like whether we're going to take cores like you see up here in the upper part of the graph, or maybe we're going to dig a pit. Pits can give us more information, but they're very destructive, and that might have some influence on the management, what's going on in that field that we don't want to see. So a lot of cases we might do cores. We also have to think about depth that we're sampling. Oftentimes today we're thinking about sampling somewhere 75 centimeters, maybe down to a meter, because we know there are effects in the subsoil that we want to also quantify. And then how are we going to segment these samples? We might segment them by horizon, or we might segment them by depth. If we're doing modeling, if we're going to use this data later for modeling, we might do it by depth. But we have to keep in mind there's probably more heterogeneity as we start to sample across the horizons in terms of the carbon in that sample. So that's something else we have to consider. And finally, are we doing manual coring, or are we doing some type of mechanized coring? And in croplands, grasslands, we can typically do the mechanized, and that's going to be a lot more efficient. You can get out, get samples quicker, sample more sites that way. But if we're in an area with a lot of rocky soils, or maybe a forest system, we're probably going to have to think more about manual coring in those cases. A little bit here, as I mentioned, I'm going to talk a little bit about methane in tool as I go through this talk, and one thing is about how we measure methane in tool. And there's 2 general approaches that are used most often. One's a chamber method, and we can use a chamber method where we're manually collecting the sample in a syringe, or we can use a more automated system like you see down here in the lower left-hand corner. And the other approach is using flux towers. Some of you may use flux towers yourself, so that's a really good way to measure these trace gas emissions as well. The application of these methods is with these chambers, you're measuring the flux at a fairly fine time scale. They're highly suitable in studying treatment effects. And the reason I say that is because a lot of our experiments have these plots laid out. You put an eddy covariance tower in there, well, you're kind of mixing all that together. You can take a chamber in and drop it into those treatments and get an estimate of the flux for each of them. And these flux data can be used to verify, calibrate process-based models. Eddy covariance, we're measuring more at a whole farm or at least whole field scale. And that'll give us more of a, think of it as an ecosystem, the whole transfer, the flux that's going on in this system. Very valuable information. It can respond, it can give us information about responses to environmental conditions and land management at the whole field scale. And again, these data can be used to verify, calibrate our process-based models. Cost-wise and requirements here, well, all we need for the flux chambers is the chamber, gas sampling supplies, and an analyzer. Costs generally maybe go up to about $50,000 US to do this. Less labor requirements there. But when we go to the flux tower, these are more sophisticated instrumentation, more expertise is needed, and costs go up, maybe over $100,000 depending on the system. This is more labor intensive, and you need a higher level of expertise, as I mentioned. Also, when we get down here at the bottom, we think about data accuracy and management. Errors can be large when we're using these chambers. Gas sampling, the detection, linearity assumptions of the gas concentrations, all that's going in this. And although we can estimate for this the effect of the treatments, there's also effects of environmental factors that are changing day to day. And oftentimes when we do chambers, we're not out there every day. We may go out once a week and take these chamber measurements, and only for a part of the day. Now, there are systems today that are more automated, which can take measurements more continuously so that we can reduce some of those impacts. But most of the time, we have a less continuous time series generated by these type of approaches. Whereas the eddy covariance, there are some assumptions about the flux calculations. There can be errors due to gap filling correction factors that are being used, but there's less of an environmental impact. When we take these chambers out, you can see here, there's an impact. We've got this chamber set up here. This is impacting the system when we're taking the measurement. That's not the case with the eddy covariance tower, or at least not as much of an impact. And we can get more continuous time series often with these eddy covariance measurements. Okay, just a little bit about sample preparation and laboratory analysis. I'm again going to start with soil carbon, and I'm not going to go through this whole diagram with you, but this is a dry sieving technique. Some of you may have done this if you're soil scientists. Basically, when we get back to the lab, we're going to take this sample and we're going to sieve it through an 8mm sieve. And basically what we're trying to do is get rid of the roots and the plant material and get rid of the rocks. And that's what we're separating out on the left and the right side here. Once we get through the 8mm fraction, we're going to sieve a part of that with a 2mm sieve. And that's where we really get down to what we consider soil organic matter. That'll contain the carbon that can stay in the soil for longer periods of time. And again, we're going to separate out the plant fraction and any rock that we find there. We may want to extend this and do wet sieving, another fractionation method where we take that 2mm sieve soil, we disperse it, and once we disperse it, this is trying to separate out what's mineral associated, as you see over here, versus what's palm, because the palm tends to float. It's a little bit lighter density. And we use this 53 micron sieve to separate those two. And the reason we might want to do this, as Hannah alluded to in her talk, is there's differences in these types of organic matter. The palm is typically unprotected or in large aggregates. It's more plant-derived material with a faster turnover time, higher, more variable C to N ratios, lower delta 13C, and it does not saturate. That means we can add palm to soil. We can continue to add that to the soil over time. It's not going to reach a level where it's saturated in the soil. And as Alan pointed out, palm does get broken down more and becomes mineral-associated organic matter. It can become over time. That fraction is protected on mineral surfaces, has a slower average turnover time. This is where we're getting that carbon that'll stay around for decades to centuries. It's consistently lower C to N ratio, higher delta 13C, and it can saturate because there's only so much surface area on these minerals. You can eventually get to the point where you can't bond more of that organic matter onto those surfaces. Not going to say much about laboratory analyses. Of course, this is the next important step. The traditional approach that we've used in the past few decades is a seed analyzer if we want to analyze the amount of carbon and nitrogen in those soils. If we're going to analyze the methane into oil, we're going to use a gas chromatograph. You see there on the right, that's an example. We could spend the rest of the day talking about these methods, and there are other approaches as well, but I'm going to move on today because this is not necessarily my area of expertise. There's probably others in the room who could say more about these instruments. Finally, calculating our soil organic carbon stock changes, greenhouse gas emissions. If we've done this sampling in an appropriate way, we can use basic survey statistics. Statisticians have known this for years. These statistics have been out there. We can calculate our change in soil carbon over time using these statistics. For example, up here, we have our delta SOC. Maybe we sampled in 2020. Maybe we took a sample back in 2010. We can subtract those. We calculate those soil organic carbon stocks based on the amount of organic carbon that we got off of our C analyzer. We need to take a bulk density estimate and the depth of the soil, and then we need to subtract off what rock fraction there is in our sample. And then we multiply that by a weight. That weight comes from the survey design. And that just depends on how you set up that design. That weight allows you to go from that sample to the entire population that you want to get an inference on, that you're trying to estimate these soil organic carbon stock changes. The advantage to this design also is we can estimate the variance. This is for a stratified design, this particular equation, but these are all standard equations that you can find in textbooks. And from that, we can calculate a 95% confidence interval. And now we know from this sampling we've done what the likely estimate of soil carbon is within that level of confidence, in this case, a 95% confidence interval. And the similar approaches could be used for methane and N2O. Very straightforward, but very expensive and rarely done. This is something that we'd like to see more measurements being taken, but it's often cost prohibitive. We don't have a lot of measurements. And so we turn to model-based approaches in most cases. I would argue more than 99% of soil carbon inventories are model-based. Now, they're informed by measurements, but they're not purely measurement-based, the approach that I just described to you. So model-based approaches, you've got to think about your model. What model are you going to use? Are you going to develop a new model? Excuse me. You've got to calibrate, evaluate that model. You've got input driver data that you need. And finally, you need to apply that model. I'm going to talk about all these things in a moment. Give me just a moment here to get a little drink of water. So there's many different types of models. I'm not going to talk about all of them today. There's empirical models, machine learning models, ecosystem models. And today I'm going to focus on ecosystem models. Those are the types of models I use in my work. And this is the Sentry or DayCent model that Dave mentioned in his talk this morning. This is kind of one of our classic models that we can use. It's not a perfect model. It doesn't perfectly estimate soil carbon stock changes or greenhouse gas emissions, but it is a useful model. And that's why we use it in a lot of operational systems. And it's an ecosystem model because it's modeling all the key processes occurring in an ecosystem, whether we're looking at a cropland, a grassland, or forest. We model the plant production up here at the top. And we're looking at these material flows in this case. You can look at energy flows too, but we're looking at material, carbon and nitrogen flowing through this system. When those plants die, you get residues. Those residues then are transformed by the microbial organisms, forming active, in this model, what's called active, slow, and passive soil organic matter. And those are kinetically defined pools that have different turnover rates. At the same time, we're modeling water flow through the system and the heat, the thermal regimes in the system, and their influence on these processes. Each of these boxes has an equation or set of equations that are determining the amount of carbon and the turnover of that carbon and then transfers onto that next pool in these arrows you see here. And this is just to illustrate kind of how that's being calculated. If we have flow maybe from this soil organic matter pool A to B, we have this equation here where there's an amount of carbon in the pool A. There's a decomposition rate constant for that pool. There's a climate effect, that's your moisture and temperature effect, a pH effect, and if it's a cultivated soil, there's a cultivation effect on that pool. And this is determining that flow, and then how much of that carbon eventually makes it to that next pool depends on the carbon use efficiency of the microbes. And that's, in here, it's a fraction of CO2 that's lost due to heterotrophic respiration. And if the microbes are more efficient, more of that carbon will be transferred onto that next pool. They're going to be converting more of that into their products and into their biomass. If they're not very efficient, more of that will be emitted as CO2. A lot of parameters involved here in each of these calculations. And some of those we can approximate from the literature, but some of them are unknown. We can't approximate them directly from the literature. So that takes me to the next step. We need to calibrate this model and evaluate that calibration. And again, there's a variety of ways of doing this, like parameter optimization methods, for example. PEST is a common package that people use for that. But there's also other approaches like Bayesian, and that's the one I'm going to discuss now. We have prior distributions on these parameters, and those may be based on the literature. They may be based on some expert knowledge, our knowledge about these processes. Some of that distribution represents reality, and that's represented by these green dots here. But some of it doesn't, and that's the gray dots, if you will. But we don't know this before we calibrate the model. We don't know which parts of the distribution are really represented and which are not. To get at that, we take observations. This is where observations, even if we're not doing a measurement-based inventory, those measurements are really critical for our modeling. We can take those measurements, run the model with all these different parameters, or parameter sets, and we compare it to those measurements. And the closer those are to each other, the more likely they are to pass through this filtering process. We use an algorithm called the sampling importance resampling algorithm for this. And they'll end up on the other side there in our posterior distribution. And you can see we might end up with a couple of grays, but we're going to end up picking those parameter sets that more closely match the observations using this process. And this is just showing you from some work that we did, the posterior distributions. These dashed lines represent the priors, and these gray shaded areas represent the posteriors. And some of them were not that informed by the data, like down here the CO2 effect was not informed much by the data, but up at the top, the temperature effect on decomposition was informed quite a bit by the data that we had in this analysis. The nice thing about the Bayesian approach is that now we have distributions around these parameters that we can use for error propagation to get that 95% confidence interval that we're trying to work towards. We also need to evaluate that calibration. This is modeled soil organic carbon on the x-axis and measured on the y-axis. And if the model was perfect, it would fall on that dashed line there. Of course, it's not. And the calibrated data set looks better than the out of sample. That's not surprising. It's a bit circular, right? We calibrate the model, and of course, it's going to match more closely to those observations. But even the evaluation data set looks pretty decent, particularly when you consider the prediction error, and that's those light gray lines you're seeing there. Most of those prediction intervals do include that one-to-one line. So given the error that we have, we are matching the observations in this case. We also need input driver data, and I'm going to go through an example to help you think about what that is. When we're using a model like Descent, we need to simulate events occurring over the growing season, like when tillage is occurring, when planting's occurring, any fertilizer events. We need to simulate when harvest is occurring, manure amendments, cover crops. All these things need to be simulated over the course of that annual cycle in order to capture their effect. And we need this for every year in our time series. So how do we get to this? Well, the first thing is, in our case, we use survey data, the National Resources Inventory. There's about 500,000 locations that are in agricultural lands in this survey conducted by USDA. And about 200,000 of those are in cropland. And so for each of those, and this is zooming into an area there in southwest Minnesota, where you have a primary sampling unit in that survey, and then within that primary sampling unit, you have secondary sampling units, and one of those is a crop field there in the lower left-hand corner. We're going to simulate that crop field, and we need all of this information in order to drive that simulation. We need to know the land use, the crop, and the irrigation. All that comes from the NRI, so we don't have to look any further for that. But the rest of this information comes from other sources. And to start with, we use MODIS Enhanced Vegetation Index. So we're pulling some remote sensing data in here using a light use efficiency calculation originally developed in the CASA model. Some of you may be familiar with CASA. We use PRISM weather data set produced by Oregon State University, get our weather data. And we're using soils data from Sergo. That's NRCS's product. For example, this soil might be a clay loam. So these geospatial data sets give us information that's valuable for doing this simulation. We also need these other management data, and that comes from a whole series of different surveys, and I don't have time to talk about all of these today, but we're basically combining surveys that go from different parts of our time series, different parts of the spatial domain, using statistical machine learning methods. And ultimately that gives us planting date, harvest date, tillage, fertilization, manure amendments, and cover crops. And we do this, in our case, 6 times. So we have 6 imputations, because there's uncertainty in this imputation process where we're imputing the missing data for this NRI survey. We don't know this on the NRI, but we use these other surveys then to impute it. And we do that 6 times to represent the uncertainty. And this shows you some of those results here. This is the fraction of cover crop in the late 2010s and the nitrogen fertilizer being applied to croplands from those analyses. And these are the averages across those 6 imputations. Finally, we need to apply this model. We use a Monte Carlo simulation approach because we want to propagate the error through. We have uncertainty in the management data, the driver data, if you will. We have uncertainty in our parameters, represented by those posteriors from the Bayesian analysis. There's also, because this is also a survey, the NRI is a survey, there's the replicate weights represent variability in those weights that are being attached to each of those survey locations. And ultimately, we select a value from each of these, run the model, select another value, run it again, another value. We do this 1,000 times. We have 1,000 results at the end, and that gives us, we use the median and then the 2.5 and 97.5 percentiles out that to get our 95% confidence interval. So I just want to wrap up here thinking about opportunities for reducing uncertainty. I think this is really critical when we're thinking about measuring. We can model these things using a model like DACEN or ROSC or DNDC or a whole host of other models out there, but they all have a lot of uncertainty, and we want to work towards reducing that uncertainty. There's several opportunities I want to highlight for us to think about this week. One is expanding our observations and our measurements. These are the long-term experiments we have that we use in the National Inventory Framework. Methane's in the dark red, you got N2O in the blue triangles, and soil organic carbon in the green circles. Seems like a lot, right? But it's not really that many. It's pretty small for what we're trying to do. This needs to be expanded, and in fact, in the Inflation Reduction Act, there's specific money there to expand this network, and I know USDA is working on this, and we're hopeful that we'll have a lot more observations in the next 5 to 10 years. And also, they're working on expanding their network of N2O and methane emissions data collection. The second opportunity is advancing our understanding. So this is a paper from a few years ago by Lehman and Kleber, where they were pointing out that given the science that we've been doing over the past couple decades, we're moving from this classic model. This is how we thought about soil organic matter decades ago, as building humic substances in the soil, humic acids, fulvic acids in human. But as we study this more, we realize these didn't really occur in the soil. They occur in a test tube in the lab, but don't really occur in the field. That really what's going on is more of a soil continuum model, as they point out here, where this plant material is being broken down into smaller monomers over time, and we're producing these microbial products, and those are being pulled onto mineral surfaces as mineral-associated organic matter, and the POM can get into aggregates and stored there as well for longer periods of time. We need to continue to advance our understanding. And from that, we can build better models, improve those models. We need to operationalize the new generation of models. There's a whole suite of new models out there. They're not quite ready, but they're getting closer to being ready to operationalize in systems like we do for the National Inventory. We need to incorporate new driver data, possibly remote sensing products. I know we'll talk more about that this week. And maybe evaluate AI as a way to inform this process as well. I know there's research going on about that. I think we'll hear about later in the week. This is a model that we work on at CSU called MIMS, which you can see as opposed to Century or DACEN has a lot more pools here in the bulk soil. And it has a rhizosphere. So it's a more detailed model based on new understanding we have about soil organic matter dynamics. And finally, improving our management practice data sets. We need to develop new surveys, use remote sensing data products to fill gaps in management data. I'm sure we'll hear more about the remote sensing part of this later in the week. So we have better data, more continuous time series of data to drive these models and evaluate the changes that we're getting. So I'd just like to thank you for your attention.